Dear friends, we will discuss some of the 2 marks question terminology. Sirtuin genes. It protects the genome by preventing wrong expression of the genes. It encodes histone deacetylation enzyme. As we know that deacetylation of histone means inactivation, and it is required for repairing chromosomal breaks. Sirtuin genes are important defenses against premature aging also. So sirtuin genes have some functions in preventing wrong expression of the genes, and these are against the premature aging. SKN1. The oocyte of the nematode C. elegans has a maternally expressed transcription factor which controls the fate of EMS cells, stem cells. It is one of the cells of the 4-cell stage that marks the ventral region of the developing embryo. Slit proteins. Chemorepulsive extracellular matrix protein. The slit proteins involved in inhibiting migration of the neural crest cells and in controlling growth of the commissural axon. SOX9, very important gene. It is an autosomal gene, very important, involved in the bone formation and in other developmental processes too. SOX9 induces testis formation, and XX human with an extra copy of SOX9 develop males. Telogens, the resting phase of hair follicle regeneration cycle. Tip cells, these are endothelial cells responding to vascular endothelial growth factor VEGF and begin sprouting to form the new vessel during angiogenesis. Crown roots, adventitious roots that emerges from the lowermost node of a stem. It means the nodes which are very adjacent to the roots. Trefoil stage. It is the stage in some spirally cleaving embryos in which a particularly large polar lobes is extruded at first cleavage. It is in spiral cleavage and giving the appearance of the third cell forming before the polar lobe is reabsorbed back into CD blastomeres. Type A is spermatogonia. In mammal, sperm cells that undergo mitosis and maintain the population of type A spermatogonia while also regenerating type B spermatogonia. Vent4. In mammal, Vent4 is involved in the primary sex determination, kidney development, and the timing of the meiosis. Vent4 is expressed in the bipotential gonads but not traceable in XY gonads destined to form testes. So Vent4 is for femaleness. However, it is present in the XX gonads becoming ovaries. Vent7a. It is an important specifying dorsal ventral polarity in the tetrapod limb. It is expressed in the dorsal but not ventral ectoderm of the limb birds. If expression in this region is eliminated, both dorsal and ventral sides of the limb form structure appropriate for the ventral surface, such as the ventral foot pads on both surfaces of the paw. Interkinetic nuclear migration. The nuclear movement within certain cells when they go through the cell cycle. For example, in the germinal neuroepithelium, in which nuclei translocate from the basal end to an apical end of the cell near the ventricular surface, where they undergo mitosis, after which they slowly migrate basally again. Kupffer vesicle, transient fluid filled organ housing the cilia that control left-right asymmetry in the zebrafish. Anaphase A, early anaphase in which the sister chromatids separate and begin to move towards the opposite pole. Anaphase B. In the late anaphase, the polar microtubules slide past each other and elongates to push the spindle poles farther apart. Simultaneously, the sister chromosomes are pushed to their respective poles. So in anaphase B, there is a movement of chromatid as well as the poles. AHP genes, Arabidopsis histidine phosphotransfer gene. AHP genes are involved in the cytokinin signal propagation from the plasma membrane receptor to the nucleus. AGO, argonaute, a catalytic protein that is part of the RNA-induced silencing complex, ARP2/3. It is the actin-related protein, second and 3, which binds to the side of the pre-existing actin filament and form a complex with the actin to initiate growth of actin filament branch. Boundary medistems, a transitional zone that separates the leaf primordia from the shoot apical meristem. Bowen ratio, from the Theisen-Ziegler book, the ratio of sensible heat loss to evaporative heat loss, the two most important processes in the regulation of leaf temperature. So there is comparison of Bowman ratio for the C3 and C4 plants. Callose, a beta-1,3 glucan synthesized in the plasma membrane and deposited between the plasma membrane and the cell wall. Callose synthesized by sieve elements in response to damage, stress was part of the normal development process. CCAAT box, a sequence of the nucleotides involved in the initiation of transcription in eukaryotes. CESA, cellulose synthase A, multigene family of cellulose synthase found in the all land plants. Constitutive photomorphogenesis, COP1, a constitutive repressor of photomorphogenesis that interact with the photomorphogenesis promoting factors such as HY5 to promote their degradation via ubiquitin proteasome pathway. COP1, so suppressor of phytochrome A. COP1 SPA complex, a protein that forms a complex with COP1 and repress the photomorphogenesis. COP9 signalosome CSN complex, a protein complex that appears to facilitate COP1 entry into the nucleus. Okay, so I think these terms will help you in solving some of the question of 2 marks. So important one is COP1, constituent photomorphogenesis 1, and it is second COP1 suppressor of the PHYA. It is the SPA complex that forms a complex with COP1 and repress the photomorphogenesis. Okay, please check these terms and I think it will help you in solving the questions. Okay, good luck.